Assalamu alaikum. Today we want to continue this chapter 19, section 19.5, effect of concentration on enzyme activity. As we said, an enzyme catalyzes reaction by lowering the activation energy of the reaction they catalyze, and by doing this they increase the rate of the reaction up to 10 to the 20. The reactant is called substrate. So substrate must first bind to the enzyme, as we showed you last time, by the Luck and Kay model or the induced fit model. Now we want to talk about the effect of this substrate concentration. Now if it is low, not all the enzyme molecules are in use. The rate increases with the concentration of substrate as more enzyme molecules are put to work. So at the beginning, as the substrate concentration continues to increase, the rate levels off as more and more active sites are occupied. So at the beginning, direct relationship, the reaction rate increases with increasing substrate reaction, then it levels off here and the rate approaches maximum. So here we have maximum and constant reaction rate. Now, once the enzyme is saturated, increasing substrate concentration has no effect. The rate when the enzyme is saturated is determined by the efficiency of the enzyme, the pH, and the temperature. Enzyme and substrate molecules moving at random in solution can colloid with each other at a rate of about 10 to the 8 collisions per mole per liter per second. A few enzymes actually operate with close to this efficiency. Now, what about the effect of enzyme concentration? It's possible for the concentration of an active enzyme to vary according to metabolic needs. So, as long as the concentration of substrate does not become a limitation, the reaction rate varies directly with the enzyme concentration. So, a plot of reaction rate versus enzyme concentration is a straight line that passes through the origin, and we must have excess substrate. The Michaelis-Menten Kinetics model for non-allosteric enzyme. Now, rate of an enzyme catalyzed reaction or the velocity at various substrate concentration. If we plot this, we will get this hyperbolic type of curve. E plus S to produce ES. We have K1, K inverse 1, and then ES going to give E plus P, and we have K2. Now, K1 is the rate constant for ES formation from E plus S. K inverse 1 is the rate constant for dissociation of ES to E and S. And K2 is the rate constant for conversion of ES to P. لاحظوا here at the beginning, velocity increases with substrate concentration in a linear way. So here we have a first order kinetics because the rate depends on concentration of substrate. But when it levels off, and as we said before, at high substrate concentration, the rate does not change and levels off, and here we have maximum velocity. And here we have zero order kinetics. The rate does not depend on concentration of substrate. This is true for all enzymes that obey the Michaelis-Menten kinetics. There is an equation called the Michaelis-Menten equation. This is the one that we plot here. Velocity versus substrate concentration, as you can see. Now, at very low substrate concentration, first order region, as I showed you, velocity increases linearly. Very high substrate concentration, zero order. Velocity is independent of substrate concentration. Vmax is the maximum velocity as substrate concentration reaches infinity. V is the upper limit of the reaction, and this equals enzyme saturation. And then we want to define also Km, the Michaelis constant, specific substrate concentration at which the reaction occurs at 1/2 Vmax. Km is fixed for a given enzyme. Actually, each enzyme has a specific Km for its substrate. Higher values of Km means lower substrate affinity. So Km is a measure of the affinity of the enzyme for its substrate. The higher the value of the Km, the lower the substrate affinity. Or the lower the Km value, the higher is the substrate affinity. Now what about effect of temperature on enzyme activity? لاحظوا again reaction rate versus now temperature. Rates of enzyme catalyzed reaction do not increase continuously with rising temperature. At the beginning, as temperature increases, the rate increases. Rates reach maximum here, maximum rate, and then decrease. Now enzymes at this point, they denature when non-covalent attractions between protein side chains are disrupted, destroying the active site. So this is the effect. How about effect of pH on enzyme activity? The catalytic activity of many enzymes depends on pH and usually has a well-defined optimum point at the normal buffered pH of the enzyme's environment. Most enzymes have their maximum activity between pH values of 5 to 9. Eventually, extremes of pH will denature a protein again. So high and low pHs denature the enzyme. Now, if enzyme regulation, feedback, and allosteric control. Now, a variety of strategies are utilized to adjust the rates of enzyme catalyzed reactions. Any process that initiates or increases the action of an enzyme is an activation, whereas any process that slows or stops the action of an enzyme is an inhibition. Several strategies usually operate together. Feedback control occurs when the result of a process feeds information back to affect the beginning of the process. Here we have a series of reactions, A going to B, B going to C, and C going to D, with 3 enzymes, 1 and 2 and 3. Now, if D inhibits enzyme 1, the amount of A and B synthesized decrease. No, the amount of B and C synthesized decreases when no more D is needed. When more D is needed, enzyme 1 will no longer be inhibited. This is called feedback inhibition. Then, when D reaches a certain concentration, it goes all the way and inhibits enzyme number 1, and this is called feedback inhibition. Now, there is an enzyme which undergoes this mechanism of control called aspartate transcarbamylase. It is abbreviated ATCase. It undergoes feedback inhibition. It catalyzes the condensation between carbamyl phosphate and aspartate to produce carbamyl aspartate, and then several steps or reactions to give the final product called CTP. What is the importance of this reaction? CTP is required for RNA and DNA synthesis. Now, when concentration of CTP reaches certain value, it goes all the way and inhibits ATCase, and this is called feedback inhibition. So ATP is an inhibitor of ATCase. Now also it's called end product. The end product in the sequence of reaction inhibits the first step in the series. Then feedback inhibition, you can call it also end product inhibition. This is an efficient system. Why? Because the entire series of reactions can be shut down when excess of final product exists. You can shut this, you can shut this, you can shut this, and preventing accumulation of unwanted intermediates like here and here. So this is an efficient mechanism. Also, we have the so-called allosteric control, the binding of a molecule. It's called an allosteric regulator or effector at one site on a protein affects the binding of another molecule at a different site. So allosteric control can be either positive or negative. So here, مثلا, you have your enzyme. This is the active site. This is the allosteric site. Then allosteric enzymes, they have, in addition to the active site, they have a site called allosteric site. To this site binds an activator or inhibitor. So in the negative allosteric control, when the negative allosteric regulator binds, it prevents substrate from binding. معناته substrate cannot enter here. This is the negative allosteric control. However, in the positive allosteric control, here we have unavailable aslan active site. When this positive allosteric regulator binds, now substrate can enter at the active site. Now, when we plot the reaction velocity versus substrate concentration, in this case aspartate, as I showed you in that reaction, we find it gives us a sigmoidal type of curve. As you can see here, we have a sigmoidal type of curve. In this case, no ATP or CTP. Now, in the presence of the inhibitor called CTP, the curve is shifted to higher substrate concentration and becomes more and more sigmoidal. However, in the presence of activator like ATP, the curve is shifted to a higher reaction velocity at low substrate concentration, and the curve becomes less sigmoidal and more hyperbolic. Now, enzyme regulation, inhibition. We have two general types of inhibition, reversible and irreversible. In the reversible, we have three types: competitive, non-competitive, and uncompetitive. Now, in the reversible non-competitive inhibition, as the name implies, reversible, it can be reversed. The inhibitor does not compete with the substrate in the non-competitive inhibitor, does not compete with the substrate for the active site. It binds to the enzyme substrate complex so that the reaction occurs less efficiently or not at all. In the reversible competitive inhibition, however, competitive inhibitor binds reversibly to an active site through non-covalent interactions, but undergoes no reaction, preventing the substrate from entering the active site. So in this case, as the name implies, both the substrate and the competitive inhibitor are structural analog of each other. When one binds, the other cannot bind, and vice versa. In this inhibition, the competitive inhibitor binds only to the free enzyme, whereas in the non-competitive inhibitor, it can bind either the free enzyme or the bound enzyme or the enzyme substrate complex. Now again, this is how the non-competitive inhibition, the competitive looks like, and this is the non-competitive. Note here, the competitive inhibitor binds at the active site exactly because it has the same conformation. So once the competitive inhibitor binds, the enzyme becomes inactive. However, they found that increasing substrate concentration can relieve this type of inhibition. Whereas in the non-competitive inhibitor, there is the non-competitive inhibitor binds to a site other than the active site. So once it binds, it changes the conformation of the active site, so the substrate can no longer bind. Or if it binds, reaction will not occur. So in this case, however, increasing substrate concentration will not relieve this type of inhibition. And again, this is the equilibrium. Here we have the enzyme. In the presence of substrate, it forms ES and then product. In the presence of I, it forms EI and no product because it's inactive. So the competitive inhibitor binds to the free enzyme. In the other type of inhibition, the non-competitive, this is non-competitive. The inhibitor binds either to the free enzyme or to the bound enzyme, as I said. So this is not the same as this. This must be non-competitive. Now the second general type of inhibition called irreversible. Here the enzyme's reaction cannot occur because the substrate cannot connect with the active site. Many irreversible inhibitors are poisons as a result of their ability to completely shut down the active site. And usually a covalent bond is formed between the inhibitor and an amino acid residue at the active site so that the enzyme becomes inactive and irreversible. That's why they are sometimes called suicide substrate or suicide inhibition. Now, heavy metal ions such as mercury and lead are examples on irreversible inhibitors that form covalent bonds to the sulfur atoms in the SH groups of cysteine residue. Suppose an enzyme that has an SH residue at its active site, cysteine residue, then these heavy metal ions will form covalent bonds to these heavy metals like mercury and lead. They form a covalent bond to the sulfur atom here, making the inhibition irreversible. Now, enzyme regulation, covalent modification, and genetic control. Let's first define the term called Zymogens. Zymogens are inactive enzyme precursor that can be irreversibly transformed to an active enzyme by cleaving of covalent bonds or splits of part of the molecule. Examples of zymogen include trypsinogen, chymotrypsinogen, proelastase, precursor of enzymes that digest proteins in the small intestine. These enzymes must be inactive when they are synthesized, so that they do not immediately digest the pancreas. So they are synthesized and stored in the pancreas. But once they are secreted into the intestine, they undergo this cleavage. So if this is your inactive form or the zymogen, notes here, this is cut here. This is inaccessible active site, and now it becomes an accessible active site, and the enzyme becomes, the zymogen becomes active enzyme. Then cut off part of the molecule, as is the case with chymotrypsinogen here. Then our example, chymotrypsinogen, is inactive zymogen. It consists of a single polypeptide chain with 245 amino acid residue. Now, when it is secreted into the intestine, it undergoes cleavage at arginine 15 here by enzyme called trypsin, because trypsin cleaves at the C terminal of arginine and lysine. Then you have here this break here, and now it's converted to pi-chymotrypsin. Although it is an active enzyme, but it is not the final form. This pi-chymotrypsin undergoes self-digestion at the following position: at leucine 13 here to give this dipeptide, serine arginine, and at tyrosine here 146 and asparagine 148 to give this dipeptide, threonine asparagine. So now pi-chymotrypsin is called alpha-chymotrypsin, and this is the final active form. So whenever you read the word chymotrypsin, it means alpha-chymotrypsin. Another point here, لاحظوا at the beginning we have the so-called disulfide bonds here, the ones in yellow. We have 5 of them. Now at the end we have still the 5, but now of two kinds. We have inter-disulfide bonds. They connect each two fragments together, and the intra, 2 in one fragment and 1 on the other fragment. Intra means within the same fragment or within the same chain, here between two chains. So we have 2 inter-disulfide bonds and 3 intra-disulfide bonds. لاحظوا here after the first cleavage here at isoleucine 16, the amino group here with positive charge forms an ionic bond with aspartate 196 residue at or near the active site, and this is important for the activity of the enzyme, whereas in the chymotrypsinogen there is no such ionic bond. That's why it does not bind the substrate. This is the explanation. Now, Covalent modification. Another mode of covalent modification is the reversible addition of phosphoryl groups to serine, tyrosine, or threonine residue. Kinase enzymes catalyze the addition of a phosphoryl group supplied by ATP. This is called phosphorylation. Phosphatase enzyme catalyze the removal of the phosphoryl group. This is called dephosphorylation. For example, if we have enzyme called glycogen phosphorylase, it exists in two forms, B and A. A is the phosphorylated form and it is more active than the B. The phosphorylation occurs at a serine OH residue here by the enzyme called phosphorylase kinase and the requirement of 2 ATP. The opposite reaction is called dephosphorylation and this requires a phosphoprotein phosphatase. So here an example, this is called covalent modification, or another word, phosphorylation. The formation of a covalent bond between oxygen of serine residue and the phosphate group. So this is called phosphorylation or covalent modification, and it is a way of control here. Again, the phosphorylated form of glycogen phosphorylase, the so denoted A, is more active than the dephosphorylated form, noted B, less active. Also, we have something called genetic control. The synthesis of enzymes, like that of all proteins, is regulated by genes. The genetic control strategy is especially useful for enzymes needed only at certain stages of development. Mechanisms controlled by hormones can accelerate or decelerate enzyme synthesis. And here a summary of mechanisms of enzyme control. Feedback control we talked about, inhibition we talked about, zymogen activation we talked about, covalent modification we talked about of an enzyme by addition or removal of a phosphoryl group, and finally genetic control, whereby the amount of enzyme available is regulated by limiting its synthesis. This is the end of part 2. Thank you.